It's really my pleasure to introduce our keynote speaker, or one of our keynote speakers today is José López. José, he's sort of a fixture here at the Center for Blood Research and at this particular symposium. I think he's been here more than I have, actually. José comes from New Mexico, where he did his, he grew up there, I guess, and he did his chemistry and then did his medical training there and then moved north to the northwest to Seattle at the University of Washington, where he did his internal medicine training and hematology training, and then did 3 years as a postdoc in biochemistry with the famed Earl Davie. And then from there he went to California for a while, the Gladstone, and then down to Baylor, and then was recruited back to Seattle wisely to the what was then known as the Puget Sound Blood Center, which is now known as Blood Works Northwest, and where he is a really widely recognized and investigator in, in everything related to hemostasis, thrombosis, the spilling into vascular function and inflammation and in other areas. He's really made major contributions. José is the ultimate in giving back. He's been involved in all kinds of organizations at ISTH and ASH, and he's really the ultra mentor. And it's always a pleasure to interact with him. He is the perfect selection for the Earl Davie keynote. I know that Earl would be pleased to have him speak today. So, José, you're up.
Thank you, Ed. It's such a pleasure for me to give this keynote talk. Today, November 6th, is my father's birthday. He would be 109 if he were alive. But, and he died quite a long while ago, but, it's, as Ed mentioned, I spend a lot of time with Earl Davie, who became kind of another father figure for me, so kind of a special day. So thank you. And today I'll talk about work that we've been doing, not in hemostasis thrombosis, but inspired by that on sickle cell disease. First, I want to just give my condolences to the Canadian Blue Jays fan for the heartbreak that you've recently had to experience. But I'll just say kind of heartbreak goes around and comes around. The Blue Jays did it to the Mariners the week before. And so anyway, they were great series and really good. So just to review what we know about sickle cell disease, we know that it's caused by a single nucleotide mutation in a codon of beta globin, of the gene for beta globin, that converts a glutamate codon to a codon for valine. And what that does is that it changes a charged amino acid to a hydrophobic amino acid, and that allows this hemoglobin to polymerize when the oxygen tension is low. This hemoglobin also, apparently, doesn't hold on to its oxygen as well, or when it releases it, it doesn't give back, or doesn't accept, the iron doesn't accept an electron. And so you generate more superoxide and more oxidants. So there's a lot of oxidative stress. In fact, Hebel, about 30 years ago, showed that sickle red cells will produce twice as many reactive oxygen species as do normal cells, just at baseline. And that's kind of the focus. I became interested in sickle cell disease for a number of reasons. One was this paper, because most of my work has, over the last 25 years, has been on the biology of von Willebrand factor. This paper from Larry McIntyre and Joel Moak and that group in Houston showed that von Willebrand factor, the newly secreted von Willebrand factor from endothelial cells, was able to mediate the adhesion of sickle red cells to endothelial cells under controlled flow. So this was all in vitro work, and we had been doing some work in which we showed that von Willebrand factor could bind sulfatide. And there was other work by other investigators that sickle cells seem to expose more sulfatide than normal cells. And so we looked at that. And here you see on the left, so this is what this is these are washed cultured endothelial cells over which we've perfused red cells. So on the left are the normal erythrocytes, on the right are the sickle erythrocytes. And what we've done with these endothelial cells is that we've stimulated them with an agonist, in this case histamine, to secrete von Willebrand factor. And what you see these bound cells. This is kind of hard to handle, but these bound cells, the normal cells, and you see many more bound sickle cells to these what we call von Willebrand factor strings. And what those are is that when von Willebrand factor is secreted from an endothelial cell, a portion of it remains bound to the surface of the endothelial cell by unknown molecular mechanisms, and the von Willebrand factor, which is a long string-like molecule, will self-associate laterally as the strings unfold. I gave a talk about that here a few years ago and all the work that we've done about that. So it's an important process that probably is involved in mediating the attachment of not only platelets to the surface of endothelium, but also other cells, including red cells. And in this experiment, which is like 20 years old, we also showed that we could block a lot of the attachment of the red cells to activated endothelial cells with an antibody against von Willebrand factor. We went on to look at von Willebrand factor in the plasma of patients with sickle cell disease, and these were sickle cell disease patients at baseline. In other words, they weren't in the hospital, they weren't suffering from acute sickle cell crisis, and you can see on the left that what we've loaded here is a 0.5 microliter of plasma. On the right, the controls are 1 microliter of plasma, and you can see that virtually every patient had much, much more von Willebrand factor in their plasma than the controls, which is controlled pool plasma. And the highest multimers, which are the ones at the top of the gel, were much larger than the ones that we found in normal. And that's an important thing because these high multimers are much more adhesive for platelets, and they're thought to be what mediates thrombotic microangiopathies such as thrombotic thrombocytopenic purpura. We also looked at the activity of von Willebrand factor in those patients, and we could do that. Shown here is that von Willebrand factor normally contains cryptic sites to bind platelets and to bind other molecules and to be cleaved by the cleavage enzyme ADAMTS13. But when it unfolds or it's treated with chemicals such as ristocetin, these cryptic sites become exposed and they can be detected with an antibody. And what we find is that as we increase the concentration of von Willebrand factor, we get more binding of this antibody that detects those sites in sickle cell. And if we divided the slope of that increase, in the sickle patients with the slope from pooled plasma, we get this thing called an activation factor. And if we multiply that by the concentration that we measure in the plasma, we get this thing called total active VWF. We correlated total active VWF with the extent of hemolysis, and that was measured by the concentration of lactate dehydrogenase in the blood, and we got a pretty good correlation. So the higher the active VWF we had, the more hemolysis we have. And we suspect that you'll also have more adhesion and vasoocclusion. So the way that you regulate the function of von Willebrand factor is, one way is through making the very large multimers smaller by proteolytic cleavage. And that's done by an enzyme called ADAMTS13. We can measure the activity of ADAMTS13 by its ability to cut a small peptide, or in other ways, which I'll explain shortly. So in terms of its ability to cut small peptides, here you see that on the left in the blue is what we get with normals. So you can get somewhere clustering around 100% being the value in pooled plasma. And we found that in terms of the ability of this sickle cell plasma to cleave a small peptide, it in general tended to be much better than in the normal, so kind of a hyperactive enzyme. However, oops, wrong way. If we take plasma and we incubate it in something that will unfold the von Willebrand factor, in this case urea, what we get is that over time, we get a drop in the size of the multimers. But if we do that with sickle cell plasma, we get no change. It's unaffected. So what we have here is a dissociation between the ability to cut a small peptide and the ability to cut VWF multimers. And if we looked at so we can also induce the formation of those VWF strings in vitro, and then we can perfuse the plasma over that and see how much they get cut. And you see that, so we set arbitrarily pooled normal plasma at 100%, and you see that every one of these sickle cell patients was quite defective in the ability to remove those strings of VWF from the surface of endothelial cells. So there's a problem with VWF. And so we thought that it might be because the VWF, or the enzyme, is becoming oxidized. And we have shown before, and this is just looking at the section of VWF that contains the GP1B alpha binding site, that's the A1 domain, the ADAMTS13 cleavage site, that's the A2, and the collagen binding site, that's the A3. Within these three A domains, which are kind of the business end in terms of adhesion of von Willebrand factor, there are 14 methionine residues. And methionine can be oxidized to methionine sulfoxide, which changes the nature of the side chain from being hydrophobic to being more hydrophilic. And right in one of these is right at the cleavage site for ADAMTS13. That's the methionine 1606. So we did in vitro experiments. This was published quite a long while ago, looking at what effect did methionine oxidation have on a VWF function. And this is just looking at in vitro agglutination of platelets by VWF. And in the presence of a chemical called ristocetin, which helps to unfold the VWF and make it be able to clump platelets. We use subthreshold amounts, so basically at these concentrations, you can see that the control, you got almost no agglutination, whereas the oxidized was quite hyperactive. And you could see that, with shear stress, this became even a bigger, this is shear-induced platelet agglutination, that was also hyperactive by those means. So we can actually, by mass spec, investigate whether methionine residues become oxidized. And so here we just have shown is VWF from one control and a patient, and in fact, there's not a lot of oxidation in normals at these sites, but we get substantial oxidation in sickle cell disease. So just evidence of oxidative damage, in this case, to proteins. And when we looked at, we can also look at the extent to which VWF is cleaved. And that's again, the activity of the enzyme ADAMTS13. And there's an acquired disease where that enzyme is deficient. That's TTP, thrombotic thrombocytopenic purpura. And if you take this 100% as the value that you get with pooled normal plasma, you can see that the extent of cleavage in acquired TTP is quite low. But in sickle cell disease, it's also way lower than in pooled normal plasma, in this case, with the exception of one of the patients that we examined. And this patient actually didn't have homozygous sickle cell disease. It was a sickle beta plus patient. And what we find is that the extent of methionine oxidation is inversely correlated to the extent of cleavage that we detect. So again, that's a functional consequence of oxidation of proteins in the plasma. And so we're just looking at one protein, but you can already see that we've essentially induced a prothrombotic, proadhesive phenotype. We did a study looking at addition of N-acetylcysteine into the blood of sickle cell patients, actually. And the goal was to test its safety. And the idea of using this was that N-acetylcysteine is a thiol-containing amino acid that can actually break down the multimers of VWF by disulfide reduction rather than proteolytic cleavage. But it takes high concentrations, so we did a protocol where we did two doses. One was 75 milligrams per kilogram in 1 hour, followed by the same amount over 7 hours. So it's a lot of N-acetylcysteine. This is all given intravenously. And then we did a high dose, which was double the first dose. And what you see here are multimer gels here at the low dose. And what you find is that you get a little dip in the high molecular weight multimers of von Willebrand factor, and this is over time. And as you increase the dose, you get more of an effect on von Willebrand factor. Undoubtedly, this has some effect on this disorder, but that's not the only thing that N-acetylcysteine does. It's not very specific. It's not specific for von Willebrand factor. In fact, if you look at this, we looked at red cells in normals and in the sickle cell patients. And what we find, so here's the profile with glycophorin A on the bottom and size on the top. That's forward scatter. And you find a population of red cell. These are the red cells. If we look at sickle cells, and we're looking at a population here that's kind of at the same fluorescence intensity for glycophorin, but at a much smaller size. If you look at sickle cells, you find that there's a big population here that's smaller, but still has the same fluorescence intensity for glycophorin. So it's got the same amount of glycophorin on it, but they're much smaller volume. And when we give, so this 1 hour is 1 hour after we've given N-acetylcysteine, you find that these cells appear to have disappeared. And that effect persists for 8 hours and even after 72 hours, although now you can see that they're starting to come back. So what those are, we think, are that they're dense cells. So in sickle cells, the red cells become very dense, and it's a complicated pathophysiology for why that happens, but it has to do with oxidation of their membrane skeleton and activation of mechanoreceptors that allow calcium into the cell. Calcium activates potassium channels and water channels that drain the water out of the inside of the red cell and make them very, very dense. Those are not very flexible cells. So when you run, in this case, this is a Fickle gradient with normal red cells, you can see that none of them will enter the gradient, but the sickle red cells, you get cells all over, including extremely dense cells right at the bottom of the gradient. So then we give the N-acetylcysteine, and you see after 1 hour that this is starting to clear up a little bit. and then by 8 hours, you have the really dense cells, which I guess you can't reverse. They're in some kind of irreversible state, but most of the rest of them are gone. And that effect, as shown in the flow cytometry histograms, seems to persist. And so what we find is that when we look in the plasma for cysteine, free thiol cysteine, and protein-bound cysteine, that the total cysteine goes up with the infusion of N-acetylcysteine. The free thiol cysteine goes up, but the protein-bound cysteine goes down. And what we've shown is that when we infuse this N-acetylcysteine, what it does is that it kicks cysteine that's formed adducts with proteins, especially albumin, it kicks it off, and then now you have free thiol cysteine, and that very efficiently goes into red cells and other cells, and we think that's what's having this effect. So to summarize this part, we found that oxidation of plasma proteins, including VWF and probably also of red cell proteins, seems to play an important role in sickle cell disease pathophysiology. So we are very interested in being able to test this in vivo, and obviously a lot of the things we want to do we can't do on people. So we want to develop a mouse model of sickle vasoocclusion. So here comes Raymond to the rescue again. He's already rescued one project, and here he comes. So we recruited Raymond Adderley 3 years ago. And I've tasked him with, let's work on sickle cell. And so he developed a nice method here where he can visualize the microcirculation in mice. In this case, we're looking at a mouse model of sickle cell disease. And you can see that in these mice, I think this is, so this is a movie, and you can see in red are platelets, blue are leukocytes, and green are erythrocytes, that in these mice you get clumps of platelets already circulating, and you get many, many, many leukocytes rolling along the surface of the microvasculature. But in general, you get a pretty smooth blood flow of the red cells. So a model that he developed is to actually add an agonist, a PAR1 agonist that will activate the endothelial cells. That activation is acute. It will induce the release of von Willebrand factor and also the exposure of other adhesion molecules on the surface of the endothelial cells. And you'll see how different this is than what I just showed you. Now you get actually clumping of the red cells. You get a lot more adhesion of the leukocytes, and you have platelets sticking all over the place. And this goes on long enough that these vessels become occluded, completely occluded. The flow stops. And so it's actually, I think, one of the first models of complete occlusion of vessels in an animal model. And you can see here, for example, that there's a lot of platelets, there's bound red cells, there's a lot of leukocytes. And so it becomes a thrombus of many cells. And just to show you here, here's what they looked at like at baseline. You can see that there's some adhered platelets, and you have adherent leukocytes that are rolling, but after a while you get these thrombi, these occlusive clots that have platelets, they have red cells, they have white cells, and when you look at them at higher magnification, you can see there all of these cell types are in there together. In fact, you see some cells that look like they've sickled in those clots. So another thing we were interested in, and I've been interested given what I said about oxidation and the production of reactive oxygen species, was whether or not these erythrocytes activate other cells. So one of the things that this model uses cremaster muscle, microvasculature. Of course, only the males have a cremaster muscle, so we have a lot of females that are left over. So Raymond had the bright idea that he would take those cells and he would transfuse them into mice that are deficient in the VWF cleaving enzyme ADAMTS13. And the reasoning here is that those mice would be more sensitive to any kind of phenotype that we'd see. So in this experiment, this is important, so the donor red cells are labeled in green. The recipient platelets and leukocytes are also labeled. So when you look at the green channel, what you're going to see is initially no cells, but as soon as the green cells come in, then you can look at the other, you can see what happens. So you can see that, for example, I'll just do both of them. Here they come. And I taught Raymond a new phrase, and that's all hell breaks loose. And you can see that you're making very platelet rich, but there's also red cells involved here and leukocytes, and eventually you're occluding these vessels. So this is so what I'm showing you here is that the blood of a sickle cell mouse acts as an agonist to activate the endothelial cells and other cells in the recipient ADAMTS13 knockout mice. And here's just looking at pre. It looks pretty clean. Post, you get all kinds of stuff stuck there. And you'll see that these vessels become completely occluded. If you give whole blood, that's got red cells, that's got platelets, that's got plasma and plasma lipoproteins and so forth, and leukocytes. But we thought it's the red cells. So here's an experiment where the red cells are separated and washed and then infused. And I think these are open. And this is not a movie, but essentially here's what it looks like at baseline, and after infusion of the red cells, you can see that you get a lot of adhesion and occlusion of the vessels. So it's the red cells acting as an agonist for other blood cells. I think in a person or a mouse that has sickle, they've already been rendered somewhat unresponsive to this constant stimulus. And here's just some quantification. Here's with whole blood. In fact, when transfused with blood from an AA mouse or with wild-type mouse, they don't have any occlusion. They don't have anything. But in about 7 minutes, they'll occlude the vessels when they're transfused with the SS blood. And this is when washed red blood cells are transfused. It's even shorter, and probably because the concentration of red cells in this case is a little higher. But the total concentration of red cells compared to the recipient concentration is quite small. So this is not that we've transfused so much sickle blood in there that it itself is occluding. It's that sickle blood is doing something to activate the other cells. So the question was, and this is the hypothesis, this looks like something that will activate leukocytes, activate platelets, and activate endothelial cells. And of course, what does that? Things like platelet activating factor, which is an inflammatory lipid, and that's also called PAF. There are PAF-like molecules that are formed by the action of reactive oxygen species on membrane phospholipids. And here are some membrane phospholipids, okay? You have the phosphate group, and then you have the head group over here. That can be ethanolamine, serine, choline, or inositol. And then you have two fatty acid chains in the SN1 and SN2. SN2 typically are these unsaturated chains. In this case, I'm showing linoleic acid. And what's the key feature of these acids, especially omega-6 fatty acids, is the presence of these methylene groups between two double bonds. There's a single carbon, which is a methylene carbon, and that's what gets attacked by the reactive oxygen species. And eventually, you'll add an oxygen to the double bond and form peroxides. And the other thing that happens in sickle, remember I told you that this mechano sensor gets activated. That allows calcium into the cell. Calcium will activate phospholipases. Phospholipase, especially phospholipase A2, remove this fatty acid in the SN2 position to yield a free fatty acid and this species, which is called a lysophospholipid. Most lysophospholipids are bioactive. They have G protein coupled receptors and they bind other receptors such as the TRP calcium channels. So we looked in the mice, we looked for lysolipids, and it turns out lysophosphatidylserine was lower in the mice, but 3 other species, lyso-PE, lyso-PS, and lyso-PI, were higher. And the interesting thing about this is that phosphatidylcholine, PC, is on the outside membrane leaflet of cells, and the other 3 are on the inner leaflet. So it seems like the ones that are in the inner leaflet are being attacked by the enzyme probably and converted into lysolipids. We also looked at the free fatty acids in the plasma, and the DHA, which is an omega-3 fat, was elevated in the plasma. In addition, omega-6 arachidonic acid was elevated, which is consistent with those acids being removed from that SN2 position. All of these are bioactive. We also looked at the plasma of patients, and we looked at lyso-PI. This also has a GPCR receptor, a GPR55. And when we looked at control, it's a low level, but sickle cell plasma has quite a lot of this. And you see it also in crisis. And when we look at these other things, in this case, it's called 9-hydroxy, what is it? Octadecaine, diene, this and 13-hode, they're both bioactive. They're both produced either by the action of cyclooxygenases, lipooxygenases, or directly by reactive oxygen species oxidation. And they're higher at baseline, and they're even more elevated in patients in crisis. So here's the final little piece, and looking at HDL. And why are we looking at HDL? We've shown that HDL can block the self-association of von Willebrand factor into the big strands. And it's also known that HDL can scavenge inflammatory lipids. It picks them up and then carries them away to the liver. And it can pacify endothelium, so it raises the threshold for endothelial activation. And here we have an experiment much like the one that we did before, where we activate with a PAR1 agonist. And these animals have been pre-treated with HDL. And you can see that you still get some platelet adhesion, but the red cells look quite unperturbed. You still have some leukocyte rolling, but that didn't change very much. So the HDL can actually prevent the vaso-occlusion. You get a little bit of attachment, but they never occlude. So, and it's known that there are dyslipidemias in sickle cell disease. They have low HDL and low LDL. And so what we did too was that we isolated from a patient with sickle cell disease after red cell exchange HDL, and we looked to see whether or not it protected the vessels from vaso-occlusion. Here's the normal HDL, and you can see that they remain patent, but with the sickle HDL, they don't work at all. And we've done proteomic analysis, and we find that at sickle baseline, compared to control, sickle baseline, and under crisis, that we have excessive quantities of inflammatory proteins associated with the HDL, a lot of immunoglobulins, and also a lot of complement components. We're still trying to sort out what this all means. So to summarize, I've shown you substantial evidence that oxidation of both proteins and lipids accounts for many of the complications of sickle cell disease. The von Willebrand factor ADAMTS13 axis is deranged in sickle cell disease. The N-acetylcysteine through a variety of mechanisms, some antioxidant and reductive and probably other means, can alter the red cell phenotype, reducing the dense cells, and that sickle erythrocytes can activate endothelium and other cells. So sickle erythrocytes are bad for you. In fact, if we isolate that sickle HDL, it can do kind of the same thing. Normal HDL protects from vaso-occlusion, but sickle HDL may worsen it. And I just want to acknowledge all the people in the lab and collaborators, and especially Dominic Chung, Junmei Chen, and Raymond. We've done a lot of work with them. Xiaoyun Fu with her mass spec, she's a mass spec wizard, and all the people in the lab. And I have a make-believe for the Canadians. Thank you.
So thanks, Jose. That was great. Couple of I've got lots of questions, but has recombinant ADAMTS13 been used in sickle cell disease? I guess that's the first one.
There is a clinical trial. It was reported in the 2023 ASH, just the safety profile and all of that, but I have seen nothing about it since then. So I don't know what the results of that are.
And do patients with sickle trait, have you ever looked at any of them? And I know that, by and large, they're healthy, but occasionally they're not so healthy under various stresses.
We've looked at a couple of them. They react differently. They're quite different, actually. I don't know if we've done I don't think we did any of the transfusions, but we didn't find any dense cells, for example, and other things like that.
Yeah, a really interesting talk. I think I'm still just wrapping my head around this interesting overlap between something like TTP and something like sickle cell disease. So similar to Ed, have you thought about using caplacizumab to see if it knocks back this kind of hyperreactivity you're seeing? And then second of all, this is probably a dumb question, but is it just coincidence that schistocytes and sickle cells look kind of similar?
Say the last part again.
Is it just coincidence that sickle cells and schistocytes, like that you see in TTP, look similar on a blood film? Is that?
I think that some of what you see in a sickle blood smear are schistocytes. I think that there is microangiopathic hemolysis. That's my bias because they look just the same. Of course, you also have the dense cells, and they're probably easier to rupture because they're so rigid. So I think, yeah, and the first part, yes, I think you could do that. We're still not quite sure of the full contribution of the von Willebrand factor to the vaso-occlusion. Because obviously when we activate endothelial cells, other things come up. But it doesn't seem to be P-selectin because we've done that, and it does nothing. I shouldn't say it does nothing. It prevents the leukocytes from rolling, but in terms of occlusion, it doesn't do it.
There was a story about fibronectin being very important for the adhesion of the sickle cells onto endothelium. Is it still part of the pattern, or is
Did you ask about fibronectin?
Yes.
Well, we haven't tested it. I think sickle cells can bind to a lot of things. And so I think that it's quite complicated. So I think if we can, but I do think that it requires that you have some kind of activation. What we find and what's been reported before is that the neutrophils are very, very active. So they're doing all kinds of things, oxidizing proteins and lipids and things like that. So I think, I don't know where fibronectin is actually. So I don't think it's on top of an endothelial cell.
You determine the fibronectin.
Yeah, yeah. Maybe Heyu would tell me if there's a fibronectin there. Yeah, yeah.
Jose, I'm wondering about protein disulfide isomerase, different isoforms, levels. It floats around also on extracellular vesicles, which of course are abundant in this disease.
Yeah, they're really abundant.
Are we missing a link?
Well, we've recruited Moa Yang, who worked with Rob Flomenhoff, and he's a very good chemist studying the disulfide isomerases, and we're planning experiments to study it, but we don't know. And I want to say some other thing, that we think, and I showed him, I got him very excited about this, that von Willebrand factor is a target for those enzymes. Von Willebrand factor in one subunit has 169 cis residues, 8% of the whole protein. So I can imagine you can isomerize that all over the place.
Thank you.
Yeah, Jose, to my understand, von Willebrand factor as well as fibronectin, maybe pro-von Willebrand factor, they do themselves able to change the disulfide bonds. That has a CXXC motif.
Yeah, the pro-polypeptide has CXXCs. It has like 5 of them. In fact, the D1, D prime, D3 domains have also 3 or 4 of those CXSCs. They're not the, what is it, CGLC, which you have in the propeptide, but they're nevertheless the kind of consensus sequences. So I think that it itself, especially the propeptide, which we're testing, they're enzymes. They're not as potent, although they're in higher concentration in the blood than PDI.
Jose, the 2 questions. HDL, is there any correlation in patients who have high, are they protected, people who have high HDL? Do we know? Most of them don't, I guess.
Well, we don't have enough samples to test, but we would expect yes.
Okay.
However, what happens to HDL is if you have a lot of these processes going on, is that I think why they have low HDL, most of those patients, is because it picks up all these inflammatory lipids, and when it picks them up, it actually shortens the half-life because they're taken up in the liver much faster. So essentially, you have a protector there, but you just overwhelm the defenses. And so I think that if you did in some way have some really high HDL, then you'd probably be better off, but you'd still,
Eat it up.
yeah, you'd probably eat it up.
The N-acetylcysteine, the NAC, used for patients who have Tylenol overdoses, are you looking at the same concentrations? Do you know?
That's
And
where
do they
we
have
got
side
these
effects?
concentrations, yeah.
Okay.
We're basically doing a Tylenol overdose concentration, yeah, really high.